Hey, Bio2 students, Mr. Yas. We're going to start a new chapter today, biotechnology. Um, it's been in the news a lot lately. You may not have known it, but the making of the COVID-19 vaccine and many things as far as food is concerned, and crop growth and production of yogurt and cheese. There's all kinds of things that we use biotechnology for, and you can see in the words bio, the study of life, which we've been on for a while, and then technology, right? Bringing in all of our technology advancements with computers and so forth. So I want to show you a quick little video to kind of introduce this a little bit, and then we'll kind of jump in and see how it goes, okay?
Biotechnology. Life with us. Did you ever imagine a relationship between research with the zebrafish and humans recovering from heart failure? Do you know how useful some bacteria are to break down crude oil in oil spills? The answer leads to biotechnology. Biotechnology deals with living organisms and the benefits of modifying them for human purposes. Nowadays, there is a wide range of applications. For example, Laura is a 16-year-old diabetic girl. Her pancreas cannot produce the amount of insulin that her cells need to transform the food she takes into energy. She needs to inject herself from 3 to 4 times daily with synthetic insulin produced with genetically modified bacteria. The process to obtain the insulin is similar to the one used to produce the growth hormone or penicillin. But how is it possible to manipulate an organism to do what we need? The cell is the basic unit of life. Researchers are still analysing what cells do and how, and biotechnologists use this information to manipulate organisms and to develop new products. But biotechnology is not new. It was already used thousands of years ago. Humans learnt that metabolic abilities of small bacteria and fungi could be useful for food production. Wine, beer, yogurts, cheese, or yeast-rising bread you may consume regularly are the result of biotechnologies that require the use of microorganisms. The possibility of modifying microorganisms to produce new drugs started in 1953, when James Watson and Francis Crick described the structure of the DNA molecule, the Book of Life. Since then, and thanks to the work of many researchers, it has become possible to modify the genetics of organisms in order to obtain new ones with new functions. Moving aside from genetics, understanding the molecular biology of life has allowed scientists to better understand disease in a way that has clarified how many mechanistic aspects of a living cell, tissue, or organ work. These findings have allowed us to interfere with the process. For example, scientists can use biological tools such as antibodies. These are very specific proteins naturally used to fight infections that can act almost as magic bullets by recognizing the target only. Antibodies have been routinely produced in the lab since 1976 and are now a therapeutic reality to fight chronic leukemia and other diseases, such as rheumatoid arthritis. And we can go further. Now scientists understand how cells differentiate to become specialized cells, or a tissue, or an organ. For example, we can use stem cells from fat, which can differentiate into different kinds of cells, as therapeutic agents to regenerate cartilage or burn skin after an accident. Or we could try to imitate nature. For example, the zebrafish, an amazing creature that can repair its heart cells if they are harmed. If we find the genetic clues, someday we might imitate them and regenerate an infarcted human heart, or even other illnesses that today have no cure.
All right, so you can see from that video, we have lots of different ways that we use biotechnology. We've been using it not just Watson and Crick's days. That just kind of helped us move forward with it and do more things with it when we discovered DNA. I mean, they've been doing things with biotechnology for thousands of years. We even have in nature. Nature has modified things. Wheat's been modified for over 5,000 years by humans. So I think that's what we have to understand is what is biotechnology. So I kind of want to basically go over some terminology. I just kind of want to introduce some things today. I'm hoping that sound was going through, maybe not. But the terminology of biotechnology is basically what we want to go over today, and then we'll get into a little bit of how we use it, and a lot of the things that we did in gel electrophoresis in the lab next door, or virtually for you virtual guys, is technology or biotechnology is what we use, not just the machine, but how they got the DNA samples in the first place, and that's some things that we're going to know in this chapter. All right, so what I want you to do, kind of have a little different notes this time. I've already printed them off, but I want you to highlight what I have in yellow and red. Here's a good start, and that's what I have in yellow is terms, and what I have in red will be definitions, obviously. So genetic engineering is the process of manipulating the genes and the genomes, and then you throw biotechnology in there. They kind of go hand in hand. That's the process of manipulating organisms or the components for their purpose of making, here's the key, useful products. Whether that is, insulin, like you saw in the video, or food, or crops. There's lots of different purposes of biotechnology. The goal is to make useful products. We'll get into that a little bit as we kind of go. Okay. Recombinant DNA. That's DNA has been artificially made. I'm going to show you a little bit of that today, using DNA from different sources. Like the great example there from the video was we can take a human gene. I'll show you how we do that in a minute. And then we put it in E. coli, a bacteria. So we're putting a human gene in E. coli. It sounds like Frankenstein sci-fi stuff, but what we're doing is we're putting it in there because E. coli can reproduce so much faster than what we can reproduce that gene in a lab. And then we can make human insulin from bacteria and then mass produce it, and makes it cheaper, makes it more affordable, makes more of it out there for people to use that are diabetics. Okay. Gene cloning, that's the controversial ones. We know that, but that is just making identical copies over and over and over again, mass producing multiple copies. So I just kind of want to show you some terminology today, and then we'll kind of build on that. So let's kind of look at these four things today. I'll just introduce them today, and then I'll kind of show you a video on the next video. Show you a video on the next video. Sounds funny to say that, but let's break this down. So what are some tools that we use in genetic engineering? So we have these things called restriction enzymes. And restriction enzymes are like scissors. They cut out a specific location on a piece of DNA. So we got all these codes, and let's say we want to cut out a place to put a new gene in. Maybe it's a disease gene. Maybe it's a mutated gene. Maybe we want to put the insulin human gene into a bacteria. So we're going to cut out a chunk of the code from that organism. Restriction fragments are ones that we're going to put in, little sticky ends. I'll show you that in a minute. And then ligase glues it all together, and then the cloning vector carries the DNA sequence to be cloned. Okay, so let me kind of show you this. So that's a lot of wording, may not make sense to you right now. Let me show you. Okay, so let's say let's just say we want to put. Let's just use the one from the video. So I want to put in the human insulin gene. I want to insert it into a bacteria gene and mass produce it. Okay, so here's my restriction enzyme. This is my part I want to cut out. So you're going to see that there's those anti-parallel things. So DNA runs from 5 to 3 prime one way and 5 to 3 prime the opposite way, right? All right, so we go in and we cut out right here at this little section of the G, and we go over and cut out, and we just separate the code, and you can see they're mirror images, right? Here's a G, C, C, T, T, A. And we cut that out. Now we're going to bring in, here comes in the human gene, let's say this is for insulin. I want to stick that in the bacteria, okay? So I've separated the DNA. It's got these things called sticky ends, which means this is the point of attachment. And I insert this gray part into the DNA. So say the E. coli is green, the gray is the human gene. And I put this in, DNA ligase comes in and glues it together. And now I have created a new genetic code. It's still got all the E. coli, and it's still E. coli. And I'm going to make more E. coli cells from their codes. But I've taken out a piece of their information and I've inserted the human one, right? And now I can produce the insulin from a bacterial gene. As you can see, as we stick it back in there, though, it's the same coding, right? But it's a different organism, okay? So if you go back, that's what we're talking about. The cloning vector would be the E. coli. It carries a DNA sequence to be cloned, or a bacterial plasmid, as it says here. So, it's kind of interesting. And you can see how we can use this for multiple purposes, all right? So I just kind of want to introduce some stuff today. It may not all make sense to you yet, but as we kind of go along, it'll make more and more sense to you, and you'll see, oh, man, this is interesting stuff, and I can see how we can use this to make human life better. But understand something, we have to be very cautious, and we are very cautious in this country. We have restrictions on this technology because, this is great. We're making human insulin. We're using it for a useful purpose. We're saving people that are diabetic, right? But what if this gray gene is some lethal gene that we put into a virus and we release it as a biological weapon on another country? That is what we want to try to control, and that's what scares us with terrorist groups and other countries that get a hold of this technology and they use it for something bad. And you guys all know that. Throughout time, humans are pretty good at taking something that's good and making it into something bad, okay? So we have to be cautious with that, and we'll talk more about that later too, okay? So hopefully that all makes sense to you. Just want to kind of get started, so have a great day. If you got any questions, as usual, shoot me an email. All right, have a good day. Bye.